Colizzi for this talk about this very intriguing title, Navigating the Uncharted Oceans. Francesco is a chemist. He did a PhD in Bologna, in Italy. After several postdocs, he arrived to Barcelona in 2016, working at the Instituto de Recerca Biomèdica. And in 2021, he got a Ramon y Cajal Fellowship, and he decided to join our institute to apply his knowledge on biomolecular simulations on marine systems. And after the year after he arrives here, he got a permanent position at the CSIC, at the Institut de Química Avançada. So he has been here for a very short time, and he has been working and trying to apply his knowledge in our systems. So he defines himself and his research as a study of the structure, dynamics, and functions of various molecular systems using primarily biomolecular simulations. And he has worked in different fields such as drug discovery, computational chemistry, computational biophysics, and finally enzyme engineering. So he has prepared something for us, so thank you.
Thank you very much, Ramon, for the presentation and for inviting me to give you a talk. It's a big pleasure to be here. Thanks also for coming here presentially. So I have structured this presentation in this way. I will give a general introduction on biomolecular simulation, and I will try to answer this question. This is probably mostly a personal question, why I ended up doing biomolecular simulation, and also we could why it is important or why they can be useful to other people. And after introducing biomolecular simulation, I will focus on a case study. It is especially relevant for the Institute, also for my research here. Is the main focus of my research on plastic degrading enzymes. And then after discussing a little bit about this, I will try to give my personal perspective on what could be the future of biomolecular simulation in marine sciences. So I start with a very straight statement and even basic statement. Biomolecules have a 3D structure. This everybody more or less knows this. We are all aware. I remember this was my first organic chemistry book when I was studying the conformation of very simple molecules, and I was actually getting my eyes crossed to try to understand the orientation of these hydrogen atoms. Then one night, after studying many hours in the library, then I got this post-it saying, "Hey bro, I downloaded chemistry software, check it out." And so I checked it out. I downloaded. It was late 90s, and I ended up like observing this. So this turned it into this, and it was nice. I was really alone in this my room, and I got really enlightened by this rotating molecule. This has been my first ever rotating molecule. I will always feel this as my little spark that let me fall in love basically with molecular structure. And since then, since I started this rotating molecule, then molecules were not just 3D structure, but they were beautiful 3D structure. And this image, I hope, may convince you also. They are beautiful. The molecules, the biomolecules, can assume beautiful 3D structures. Each of them has a name, has a function, and it's very intriguing to try to understand how these may work together, for instance. And they are not only beautiful, but they can also be useful 3D structure. They have useful 3D structure. For instance, here you can see these are the hands of myosin interacting with, or shaking hands with actin, and together they generate the force that is needed for many movements in the muscle of the muscle. And not only we observe this actin and myosin relationship, but we can also observe that there in these muscles there are a lot of these red shapes that are molecule of myoglobin. That is the protein that store oxygen and then release oxygen to the muscles in order for them to work properly. And if we zoom a little bit on this myoglobin structure, then it will look more or less like this. This is how we visualize it in our computer. And if we compare the pig and the whale myoglobin structure, they will resemble. They will look pretty much the same. But then if you go a little bit more into details, you may notice that there are few positively charged residues on the surface of this molecule that are not present in the pig one. And 10 years ago, a study from Mirseta, they tried to understand why this was happening, and they analyzed many myoglobin structure, and what they found that this was a general feature of deep diving mammals. And what could be the biological, the functional consequence of this is that the extra charge will increase the solubility of these molecules, and as I show you here, in the muscles of a whale, you need a lot of myoglobin in order to assist the deep diving and the absence of oxygen for a long while. So, and this is just for me, it's a beautiful way to see how very little modification in the structure can basically allow very large changes in the life of a microorganism. And all right, so the next step that I would like to introduce is where do these 3D structures come from? So mostly so for the last 50 or more years they've been coming from experiments mostly. So there are X-ray crystallography, NMR experiments, cryo-EM experiments, and all the structures that have been solved of biomolecules are stored in what is called the protein data bank with more than 200,000 structures. But in the last year was very lucky. There's been a very lucky and fortunate wave of development in the artificial intelligence, in the deep learning field, and this has brought to the generation of many algorithms that can generate and predict the 3D structure of a protein with very high accuracy, similar to experiments. So this is basically this capability of predicting the 3D structure of biomolecules is now unlocking new avenues for biomolecular modeling in many areas where usually this was not supposed to be. And one of them is, for instance, marine sciences. And I will tell you a little bit more later. So biomolecules not only have a 3D structure, but this 3D structure are always in constant motion because they have thermal energy. And understanding their motion is really the way one can understand how they work. And if you allow me like a parallelism with this galloping, if you've never seen galloping, if you've never seen a horse galloping, it would be very difficult to understand how it works if you just look at this snapshot, for instance. So you need the entire picture, and to generate this entire picture, a trajectory, let's say, the motion picture, at the molecular level, there are several tools. Our preferred tools, and probably one of the tools that the community uses the most and that's developed the most, are molecular dynamic simulation. In very few words, you define a set of rules that define the energetics, the energy of your system, and then you apply Newton's law, and you integrate to resolve the trajectory as a function of time, and this allows you basically to observe the galloping of your biomolecules of interest. So one may think, okay, now we can simulate the trajectory with molecular dynamics, then we can basically observe whatever kind of biomolecular process. This is not necessarily true because biomolecular processes happen on different time scales, and for instance, those that are quite interesting for biology, for instance, ligand binding, substrate binding, enzyme catalysis, protein foldings, they happen on this millisecond time scale. It is very difficult to reach with molecular dynamic simulation because you have a very little, you can take very little snapshot of the evolution of your system, femtosecond time snapshot. So to arrive to milliseconds, you have to do this many millions of times. And how we can solve, how the community has solved this, problem to reach, observe this kind of interesting events? Well, either by building dedicated computers, or, which is a more, let's say, less expensive, less demanding strategy, to develop tricks, statistical mechanics tricks, theoretical physics tricks, theoretical chemistry tricks, to accelerate the time of exploration. And the interaction between these two standpoints, like more powerful computer and new methods that have been developed to accelerate time, is really changing and transforming the field right now. All right, I will just give you a very, I hope this is not too technical, but this is what, basically what I'm saying is that the tricks that we use, they basically allow us to, if we start from this point in what we call the free energy landscape, or all the possible conformation that our molecules like to be, the tricks that we apply, they help us to escape from this and observe different states of our system of interest. And also, if you allow me, like a wrong, probably, parallelism with marine-related things, it would be like getting the entire water column of your position, rather than just like taking one point from the bathypelagic, and you don't know what's going on the other rest of the water column. All right, so. Many applications have been developed and used of molecular dynamic simulation to answer many different problems in biology, in biophysics, in material science. We can study conformational flexibility and stability of whatever material, basically. We can also generate perturbation. One thing that is really cool about the molecular simulation is that we can control any microscopic parameter of our simulation. So there are sometimes things that you cannot do in the experiment, some condition that you would like or you are curious about, but you cannot really test experimentally for several reasons, solubility, whatever. And here you can try to face this kind of question with the molecular simulation, because basically you have a toy that you can build and manipulate in the way you want. And there are other processes that can be observed in a, for instance, dynamic process, ion permeation, again, conformational changes, protein folding, all these kind of things. And one thing that is really important, and like let's say this closes the little chapter of why biomolecular simulation are interesting, is that we can use this kind of simulation in a prospective manner. So to predict something that we don't know at the time we are doing the simulation. So there are sometimes, many times, we use them in a retrospective. There is an experiment. Someone would like to understand more about this experiment, so we run the simulation. But then one of the really advantages of molecular simulation, or simulation in general, computation in general, is that you can predict something, and then you can also reduce the amount of experiments required as a proof of concept of the hypothesis that you are generating with this simulation. All right. So now that I close this little introduction, I would like to talk about a case study that is undergoing in my small lab and concern the characterization of plastic degrading enzymes. This comes from the observation that there is an industrial and societal challenge. We produce a lot of plastic. This is not the problem itself. Well, by the way, I will focus on PET plastic. I'm not talking about all type of plastic. This part is focused on the PET, the polyethylene terephthalate plastic, which is the common plastic of plastic bottles, for instance. So we produce a lot of plastic. This is not the problem itself. The problem comes when we try to recycle this plastic, but we cannot really do it because our methods at our disposal right now are not really so efficient. Although we go every day to bring our trash to plastic. So the mechanical, for instance, mechanical recycling can be effective, but after 5 times, for instance, it has been estimated, the properties of the plastic is really deteriorated, and you cannot use it anymore and also chemical recycling is quite expensive, and then there are a lot of byproduct produced, and this is not really eco-sustainable because there are also these bioproducts that can contaminate the environment. So at the end, what happens is that many of these plastics that we produce and we release in the environment ends up in the ocean. And of course, we would like to reduce this because this is affecting many ecosystems, and this is affecting our ecosystem as well. So One possible solution that is emerging as a innovation in the plastic recycling field is to use enzymes. So basically, if this is the chemical structure of PET, you have an enzyme that can cut it a little bit and then release the bricks forming PET. And then you can collect these bricks and then put them again into an industrial process and generate plastic again. This is not like, we don't want to destroy or cancel plastic. Plastic is a beautiful material. It's very, it's of outstanding importance for our modern life. All this is plastic. So we cannot just say, well, we don't want plastic anymore. We want to do it, we want to use it in a way that we don't harm anymore the environment. So, and what is the challenge here is that we need to do this production of the bricks, the original bricks, at a cost that is convenient when compared to producing it directly from virgin material coming from oil. If we reach this, then we can turn the scenario basically into another direction. And just to give you a more convincing proof that plastic integrated enzymes can really work, I will show you this video from our collaborator at the University of Leipzig, specifically from Christian von den Decker. And you see here there is a plastic container that could be the one that you pick up at the supermarket to bring at home your fruit or whatever, and your apples. And then, so this is what happens when we put them incubated with our enzymes. So it's basically disappearing in a little bit more than 12 hours. So, all right, so it looks like, well, this is, we have already the methodology. What's wrong? Why we don't use this already? So the point is that these experiments, these enzymes are not yet available, or not yet, it is not yet possible to upscale them at the industrial level. You need a buffer, you need several conditions that you cannot simply upscale to industrial level, it will be too expensive. So there is a constant need of improving either their stability, thermal stability, so that you need less, if you go to upscaling, if you go to industrial level, you need to add less enzymes, so this also will reduce the cost. And also a way to improve the activity of these enzymes at different level. And this will improve the efficiency, so you add a little bit of enzymes, very stable, and then you will degrade a lot of plastic. This is the general idea. And so there are many papers coming out about beautiful and super powerful enzymes, but still we need a lot of effort to try to understand how to modify these enzymes so that we can use them at the industrial level. And actually, this understanding, so this the jump, the leap that we could do from a laboratory scale to industrial scale may come also from a rational design of these enzymes, so that we can really think about what are the modifications we need to get this goal. And this rational design is most of the time linked to a kind of molecular understanding how these enzymes are working. And after we understand this point, then you can start thinking about how to modify them. But first you need to understand. And historically, it has been very difficult to understand how these enzymes work. Here I wrote this sentence: the binding mode of the substrate to PTAs has been disputed for years, which means that basically the interaction of PET with the enzymes is not really clear. It's very difficult to determine experimentally. Also at the computational level, this has been very difficult to achieve. So for many years there were people, authors suggesting some mechanism, and then other authors say, no, well, this should not be like this, should be in another way around. So there were many questions raised, especially about the conformation of PET, of the polymer, and also how many subunits are needed really to bind, and so on and so forth. So when I started this project, the main question really for me was, for us, was how does PET interact with PTAses? This was an open question. So we were lucky enough with Anya, that made most of the work here, to have also a beautiful collaboration again with our colleagues in Leipzig, and we could combine our simulation with their solid state NMR experiments, and where they basically build a kind of matrix, glass matrix, where the enzymes and the polymer were trapped, but not fully frozen. They were very slowly, they could very slowly move, and this enabled to detect by NMR, nuclear magnetic resonance, to detect the possible intermediates of the reaction of this enzyme. So then it was difficult anyway to visualize in the 3D space how the mechanism how the enzyme was working. So we tried to iterate and to have a kind of a direct interaction between the simulation that provides a very specific with atomistic details the process and the data, the spectra coming from the NMR experiment. And then I would say also that during the simulation we can also manipulate the degree of crystallinity of PET. I leave this as a sentence there, and then I will come back to this again. All right, so we used several types of enhanced sampling tricks, those that allows us to explore the full water column, let's say, of the conformational landscape of a molecule. I will not go into the details of this, but we could characterize energetically these interactions. And the one thing that actually, as a result of this simulation, came out is that we only need one. So there is only one subunit. So if this is the PT, and there are several subunits interacting with enzymes, 1, 2, and 3, and 4. So only one subunit is the one that is really tightly bound to the enzymes, and all the others are quite fluffy. And so, okay, well, this is well, not such a big details, but then, like, if you to this simulation, then we add another kind of simulation where we virtually cut. Here there is no bond anymore. Here there was a bond. Here there is no bond anymore. So this is possibly the product of the enzymatic activity. Then this subunit was very stable, becomes very unstable and detached from the enzymes. And basically a new substrate can bind there and let the enzyme cut itself. Then what we observed also was that the PET chain can slide on the active site of the PTAs. So, and the sliding movement is coming from this observation. So here are two, let's say, ensemble of shapes that the PET can assume. One is the red one, and the one is the blue one. And you can see that it's basically making this kind of hopping from one side to the other. All right, so this is again perhaps not so relevant, but when you gather all this information all together, then you can end up with a kind of mechanistic insight of how these enzymes work. And this is the model that our colleagues built up. So it's a kind of processivity of these enzymes. So the enzyme enters and bind to PET, just tightly with one subunit, and then the PET can glide basically on the surface until some spontaneous detachment happens. And while it is bound, it can cut a little bit. So this information that looks like, okay, you're telling me a story, but then can be actually translated into rules that we can apply to design enzymes that may interact or may be more efficient in cutting the PET, the polymer. All right, so I would like also now to make a step back and just to give a little bit details on PET itself. So PET is a semi-crystalline polymer, which means that it has an amorphous portion. It is a mixture of amorphous PET, where you see here the chains are not really well ordered, and then there is also a little amount of crystalline PET, where there is a very high order of interacting chains of PET. And the enzymes actually can only bite and eat and access usually the amorphous portion. So the crystalline part still remains a problem. It's not accessible to the enzymes. And what people do, what is the typical approach, is to apply pre-treatments required to increase the amount of amorphous PET and also the accessibility of the plastic that you want to degrade. This can be done in several ways. But at the end, what they do is to increase the cost of the process. And I recall the first slides. We need to reduce the cost of the process, otherwise it would be not competitive to take in the plastic directly from petroleum-based materials. So now it is considered a grand challenge, even an impossible challenge somehow, to be able to develop enzymes that can target the crystalline part. All right, so how does this amorphous PET PT and the crystalline PET look like if we go again into the molecular detail? And the experiments say that basically, so this is the structure of a dimer of 2 subunit PT. There is basically this rotation that can determine whether the polymer is like this or like this. It's here. And what this plot is saying is that is basically that most of the time in amorphous PET is like this, and very little, 10% of the time, 9% of the time is straight. Okay, so why this is important? Well, because when we do the simulation, we need to respect this kind of experimental observation. We want to import this experimental observation in our simulation so we can do better prediction. And we were able to do this, to control this in our simulation. You can see here that the trans, which is the very little 10% trans to gauge ratio, this one is very little populated, and this one is populated 90%. So, and once we can handle this, then we can have a better information on how the interaction, how the enzymes can cut the polymer. So, and now we'll have one more layer, because we are playing with the conformation of the polymer. Basically, we can control everything again of our system, and we can say, okay, now we can idealize a completely trans, a crystalline PET, and it will look like this. So this is how the amorphous PET would look like if you have a very powerful microscope to look at, and this is how the crystalline part would look like. And again, the crystalline part, for instance, is the one of the neck of a bottle, of a plastic bottle. The flank is made of mostly amorphous PET, while the neck, that is very rigid, is made more by crystalline straight PET. All right, so now we can model this. Let's say, okay, let's study how the interaction of crystalline PET occur with the enzymes. So we can model again with all the tools that we developed, that we have validated against NMR, solid state NMR experiments. We can model this kind of interaction. Well, this is a kind of a pictorial view of what could be the possible configuration, a possible way of interacting. And we can also quantify energetically how much the PET like or dislike, actually the PET dislike the crystalline part. And this comes from this kind of curves. So basically what this kind of curve is, it means that the fact that, so here we are measuring, for instance, I will try to give you this detail, the proximity, how easily this oxygen, that is the oxygen from the enzymes, and it's basically the scissor of the enzyme, can reach this carbonylic group, that is basically the point where it needs to cut. So, and if they are not close nearby enough, basically if the scissor is not long enough, it cannot really cut the PET. But if this is close, then you can really cut. And this is what this plot is saying. So here the amorphous PET can get quite close. This 3 angstrom distance is relevant for this kind of cut, while the crystalline PET cannot reach this kind of proximity, at least with this enzyme that we use. And this brings us to the final slide of this little chapter about the plastic degrading enzymes. That now that we can model the interaction between crystalline PET and the enzymes, we can perhaps think, and we know and we can quantify that the enzymes does not like this kind of substrate of crystalline PET. We can think about how we can modify the enzymes so that they can interact better with crystalline PET. And because we have a way to quantify this likeness or likeness. All right. So this is just a final remark on this topic, that this computational pipeline that we are building up is not alone. We would like in the long term, this is a long run, to integrate it with like PET degrading enzymes, to analyze PET degrading enzymes that come from marine metagenomes, and then basically end up with perhaps proprietary enzymes coming from the ocean that together with partners can be developed and tested to provide a new solution for PET recycling factories and producers. But for the moment, for instance, in using PET degrading enzymes, we have observed a few limitations. For instance, the few marine enzymes that have been discovered, they have little activity compared to the terrestrial ones. So we are not really convinced that we should pursue them rather than the terrestrial ones. And also one problem of typical of metagenomics or genomics data in general is that sometimes we have incomplete sequences. So we don't know the exact sequence of the enzymes, and this is impossible then to model into a computer. Although we can model, but it doesn't make sense to model just a portion of your enzymes. The enzymes work as a wall. So what is the future? Once that I've told you all these things about the application that we have developed, what is the future, if any, in marine science? So this is a very personal perspective, I would say, from the 2 years interaction that I've had here with you. And I've identified several possible applications. So interpretation of molecular adaptation in the ocean, protein structure prediction to label the function of unknown genes and proteins from transcriptomics is something that arose in our very first discussion with Ramon. And also, we can also study ecotoxicology. We can also try to interpret and do ecotoxicological analysis. Can shed light on properties of water in general. This is a very physics-based standpoint. And then also try to extend our molecular understanding of chemical ecology. So here the weight of the, these waves just next to this possible application refers to how difficult I consider each of these tasks. So here, interpretation of molecular adaptation is the one that you have basically a tsunami here dropping on you. It's very difficult from my point of view, for what I observe, because molecular adaptation are very complex processes. It's very difficult to understand what is the molecular determinants driving one adaptation to the other. And then there are other things that can be done in kind of calm waters, protein structure, prediction of protein function. This I will show you a little bit about this. And then there are other things that are halfway in between calm water and the big waves. All right, so I already mentioned that molecular adaptation processes in the ocean are very complex processes, and also even more complex to dissect to understand what's going on, why the molecule, one specific molecule, is changing this amino acid into this amino acid from one to another. But if you look at the temperature, for instance, and if we look at the temperature from a molecular point of view, what is known is that if we take the same protein from a warm adapted microorganism and we compare the same protein to a cold adapted organism, we will see that the warm adapted organism the protein coming from the warm adapted is more rigid, is less flexible, while the cold adapted one is very flexible. And this is like kind of, okay, wow, this put us a light, should put a light in our head, because basically flexibility and dynamics is something that, well, is what I've been talking to you so far until now. And it's something that we can measure easily with simulation. And for instance, what has been observed in this work is that if we take the same enzyme, by the way, a PTase, a peptide hydrolase from a mesophilic bacteria, from an Antarctic one, actually you look at how flexible the same portion are in the two different enzymes, you will see that the Antarctic one is more flexible, has higher flexibility when compared to the mesophilic one. And this can be used as a way also to estimate whether or not there has been adaptation, a molecular adaptation to temperature factors. This has worked. This thing is coherent with this general idea, but of course this has worked for this system. Doesn't mean that it will work for all the systems that we may want to analyze. But this is a possible way to explore this kind of issue. One thing that I would like also to state and to highlight is that we do not necessarily need all the time to study the dynamics. I told at the beginning that yes, it's very useful, but well, this study just disprove perhaps what I say, but what is important about this study is very recent. They for the first time ever, at least from my understanding, they combine structural information driven and boosted by the advancement in protein structure prediction by deep learning. For the first time, they were able to combine structural information for a large amount of sequences of protein sequences with population genetics, and they use this to elucidate the selective pressure that have shaped somehow evolution of some system that they studied. And basically they found a tight association between genetic variation and protein structure, and then they also found that non-synonymous polymorphism avoid buried sites and the active site of the enzymes. And these are kind of information you cannot get without considering the three-dimensional structure of the product of your gene, which is the protein. Right, so now one few highlights that I would like to present here and to comment with you is that how structural modeling in general can be used to orphanize protein function by using something that is called, that I call the structural matching. Basically, and this is something, this kind of title is motivated by the one very first discussion that I had with Ramon, that we have plenty of cases where we found it very interesting, and we really would like to know what kind of protein is this one that is very highly expressed in our experiment. But then when we check in a sequence database, there is no possibility to identify the function of this product, even to know what it is. So I was very tempted actually to start this kind of research line, but then like the plastic degrading research line attracted me more because I saw it as a kind of more feasible with my expertise and skills. And in the meantime, in these 2 years actually, there have been many tools that have been developed, and now I just did this prediction yesterday night actually. Is that basically you can predict the 3D structure of your gene in 2 seconds. Well, now don't look at the very how if they make sense or not, this kind of prediction. But basically, you can predict the 3D structure, select a portion of your protein that is very well predicted at the structural level, and then use a structural matching, let's say, algorithm that's called SEEK, among others, to predict the function to predict the function of your unknown protein based on the structural similarities to known proteins, to proteins that have been already labeled and assigned. And actually, if we think that this is something related to phagocytosis, I think these enzymes, these proteins that are here, they may make some sense. Then we can discuss more about this, Ramon. So, and the very few things. The very few things that I would like also to mention here is that we are also in a position now to observe in a kind of real time. These are kind of very recent methods that I developed in my previous biomedical life, let's say, and but basically they allow to observe in real time the binding of a small molecule, of a ligand, of a substrate, of an inhibitor to a protein, and this binding is coherent with the experimental results that are known for this protein ligand interaction. So, and this could be like as a starting point to analyze several types of issues. For instance, link different expression profile pattern of enzymes to different substrate binding properties. So there are enzymes that may have perhaps just a few modifications in the tunnel or in the active sites residues that may interfere, and this may be related perhaps to some, may have some relation with the expression level of these enzymes, of different time in different strain or bacterial strain. So again, this kind of simulation could be used to understand a little bit more how the chemical communication is done between different organisms in the ocean. And I think also this could be used to predict the interaction of contaminants with biomarkers. For instance, we can build easily the turtle, a turtle protein and then a dolphin protein, and see whether some contaminants they can really inhibit interact with these proteins, and then do kind of a screening in a virtual screening of contaminants to whether to assess whether this interaction is possible or not possible before doing the experiment about that. All right, and this is the very last slide. So I move a little bit from the typical biological system, and I go into something that is more physical, has a more physical behavior. And actually, it was the very first discussion that I had with José Luis Pellegrini, back to those days, was the director who welcomed me to the institute. And while we were knowing each other, he talked to me about this kind of observation that he wrote, that he read. And basically, it relates to the fact that when we have a tunnel, a nanotube containing hydrogel, and we put this in water, there is a spontaneous flow that occurs apparently without any pressure gradient. So there is a flow here just because you have an hydrogel there. This has been reported several times by Pollack. And I mean, we don't know really what's going on here. And a question we are trying now to set up to answer, and there will be probably a student, a guy into students working on this topic, is if we can observe surface-induced intratubular flow with molecular dynamic simulation, with all that I've presented you so far. All right, so this is the last slide. So what is the take-home message of all this information? Is that the biomolecular simulation and modeling allow to observe and quantify and also manipulate a variety, numerous chemical process relevant to, and here you can really, I think, add your discipline and say, well, think about that. Maybe there is an application that you haven't thought about, but that could be possible. And so perhaps my last also slide, that I've been perhaps among the first biomolecular modeling guy in a marine research institute, but probably I will not be the last one. Like it's, I think so. There might be a future for biomolecular simulation. Of course, you need people that are really interested in exploring with this high-risk environment. And here comes the acknowledgements. Many people, well, the people of my more tight collaborators, Tania Di Pietro Martitelli and Alexandra Isha, together with me are what we call the Molecular Ocean Lab. Then also, of course, all the group of ecology of marine microbes, Silvia, Ramon, Ramiro, Pep, I mean, everybody really, you all have contributed to a little bit of my steps here at the Institute. And also Marco Talone from a different department, for he really also guiding me in many also practical issues. The Oficina del Supporto alla Ricerca, without the support of them, I would be really still lost and trying to find the entrance of this institute. And of course also José Luis Pellegrini to really allow me to enter the beautiful world of marine sciences. And I thank you all for your attention. I would be happy to answer your doubts or whatever curiosity you may have.
Okay, thank you very much, Francesco, for this beautiful talk. I think we have time for questions, and in fact, I'm going to start. I like a lot your opinion, this slide with opinion that you have about the future of these biomolecular simulations in the marine field. In some cases are more feasible, some cases you see it's really a big challenge. And my question is, what is your future on that? Are you going to keep working on the marine field, or you are going to move? What are your plans?
So my plans is to keep going with all the projects that I've started, which somehow touch all the points that I've shown you here, that I listed there. And then we will see now how these things work. So my idea is to keep the interaction, the collaboration with you all, although I'm moving to the other institute, and try to, continue this. If things becomes very problematic and then I don't have the resources to keep going, of course I will have to choose in a practical way which direction I will have to bring the bread at home. But it's, I mean, my idea is really that I would like to continue these things, but being here for a couple of years, I realized that some things that I thought it was doable is very difficult, actually. And some other things, for instance, the function prediction, that back to those days when we spoke, I thought, wow, this is impossible. I mean, I'm going to spend all my entire Ramon and Cajal just to find, to develop something on this. Now it has been already done by others, boosted by this deep learning.
So you did it yesterday.
Yeah?
You did it yesterday instead of
no, but I had it already there as a slide. I didn't. There was the last prediction missing, and I tested. I was really amazed that it was. I was like, well, let's see, let's give it to this tool a try. I mean, I would have spoken about the tool anyway, but it was really fast to put the sequence that you passed me 2 years ago almost. Then put it there in 2 seconds. The system generated 3 structure, and then after generated 3 structure, the 3D structure of the protein, it makes the match with a database of millions of already annotated proteins to predict the function.
So, and another question is about the, I mean, you know that in our field we always face the need to make the functional annotations of the genes that we found, or because we found in the metagenomes or we found in the expression experiments. So these functional annotations, do you think the future is doing now? Because we do it by reference sequences, basically.
Right, and I think
that's the future
like
for the annotation?
all the tools that you're using right now are being infiltrated somehow from this structure-informed information, structure-informed analysis, predictions. So, I mean, I think that it could be almost kind of included into the option that you may select when you run your typical blast to do a 3D blast. I mean, there are already 3D blast tools.
We don't do it now at all. But
that's also why
very
I
recent.
that I wanted to talk to you about this, because there are these new options that could be explored.
Hi, thanks, Francesco. It was an amazing talk. Now I'm kind of realizing I missed the opportunity over these 2 years. But so you mostly talk about enzymes. There's a reason why, of course. But this would work for other types of proteins, like membrane transporter proteins and things like that, I guess.
Yes, there are. You can run simulation of transport protein, membrane proteins, receptor, nucleic acid even, RNA, DNA, that some of them they have a very well structured shape. So the parameters, let's say the way to run a nucleic acid simulation is a little bit behind, let's say the quality of the parameters to run a protein information, because the field started probably developed more on the protein side. While the nucleic acid field has remained a little bit behind, but now there are many more advances. You can really simulate all kind of proteins. They don't need to be enzymes. This is the question. They don't need to be enzymes.
Okay.
You can even extend it to nucleic acid, RNA, and DNA. Of course, it becomes later more difficult. So there are people simulating the entire ribosome and even an entire virus. They've done simulation of the entire virus during COVID. They try to simulate the entire COVID period. So it's,
and then near the end of your talk, you showed us this development of yours of kind of visualizing or studying how the protein interacts with the molecule. Where are the binding sites and how does it work, and with many applications for that. And I'm thinking of one. So if I use very experimentally an inhibitor to inhibit the processing, the consumption of one substrate, and I know just that it works because it works, but then I also see that probably it's not 100% efficient, so it's not inhibiting all the possible ways of consumption of that molecule by microbes. And we know some of the enzymes, or many of the enzymes that mediate these different consumptions. This is to say that by your type of simulations, is it possible to say what is inhibiting what of the potential of the different candidate molecules would be the target of the inhibitor? This.
In principle, yes. I mean,
Okay.
it doesn't sound to me now with this tool in our hand, in my hand, this, not recording this, right? I mean, well, okay. So with this tool in our hands, yes, we can do it. I mean, at least we can try it.
Yeah,
Then see what happens. Of course, perhaps many of the structures that you will, many of the sequence of the enzymes of the candidate target of this inhibitor are not known experimentally, so we have to build a three-dimensional structure. There can be limitation in this also, but there is like 3-4 years ago, I would say, wow, this is very difficult. Now, we can do it. We can try. We can try to see what we get. I mean, we can see if the binding is occur, we can see really a real clear signal about the binding of the small molecule to be linked to that.
How do you, because you mentioned this applications for chemical ecology, or for the chemical talking of micro, how do you link one thing to the other? I mean, what are your ideas?
I think, for instance, so there are, I've been studying glipdepsin, for instance, which is a molecule, a depsipeptide that some tunicate produce, and it is used, actually it has been developed now as a drug for anti-cancer, and actually is under clinical trials for anti-COVID treatment. But, so what I was wondering is, so this molecule is produced, many natural products are produced as a defense, So, and I would like to, I would be curious to see, for instance, why the algae, I mean, perhaps I should go a little bit more into the details of the biology of the system to understand whether they have or not the target. Perhaps they don't even produce the target protein that is target in humans, for instance. But one thing that could be done is try to understand why the same molecule is toxic for one organism and not the other. For instance, it's communicated to that organism something and not the other. And this could be something that you can reduce at the molecular level, perhaps. Very little modification. I can send a radio signal, but if you're not tuned to capture my radio signal, you will not get it, even if you are there with the radio in your hand. And this may happen in a similar way at the enzymatic level. So if there are some mutations that make the binding almost impossible, even if you also perhaps evolved it as a way to protect from this kind of toxin. So this is something that can be done also with this kind of stimulation. This was actually one of the, coming from the biomedical field, this was one of the very first, even before the plastic degraded enzyme, this was one of the very first things that I tried to figure out that could be done. And then I put it in the
I have another question. I mean, you identified that one of the main problems when you work with metagenomes is the genes are incomplete. So how can you evaluate that? I mean, is this 3D models?
I mean, yes.
It's enough to say this is incomplete and...
Yes, so exactly. So for instance, for the PTAs, for the PET hydrolases, we try to build our structural prediction tools to start modeling this. And then we saw that there was basically an entire portion of the enzyme that was missing. The structural prediction tool can still give you the prediction, the right prediction for that portion that is present, but then when you compare it to known enzymes, you see that it's really missing. And say, wow, this is now here there is so you have just a small, I don't have, but it would be really like you can, you superpose the two structure. And then you see that there is a perfect matching for the structure, perfect matching, but then there is a much more protein that is just missing in the one.
And this is why you have a huge reference database of images, no? I mean, if not, I mean, the gene has been evaluated in the past, and also, then you have a new one, and then you see that it's incomplete.
The huge database
of structures, of genes.
Yeah,
I mean, that's the...
Now you can have this huge database because we can predict the structure of these sequences now at the basically no limit level. Okay. So they can predict structure I think that now for every sequence that has been generated, I don't know where you guys store it, but for every sequence of a genomic sequence, you have a 3D structure It will be calculated.
Okay. Wow.
And you can try to search whether your protein has some, your protein structure has some similarity with the structure of those that have been predicted.
And this has been developed in the last 2 years. Wow. Yeah. Okay. So perhaps we can leave it here. Thank you very much for this.
Just a quick question. No, just a practical question. What do you use to do this quantification? I mean, in terms of power.
It's a computer cluster. So yeah.
You don't run it on your.
No, I don't run them on my laptop. But yeah, I need. I mean, it depends. But the molecular dynamics simulator are quite expensive, and I have to go to supercomputers. But the structure prediction, there are plenty of web servers, for instance, that are available online. So you don't even need to install, or you don't even need the resource of, for instance, GPUs, or they can lend you basically the GPUs, their own GPUs to do the prediction of the structure. But the simulation, yes, you need some big computers.
Okay. So thank you very much.
Thank you.